7
Not
and
having that one on, the one right over the screen.
You want me to
Turn
take
them
that
off
off?
one by
Okay,
one.
I will turn that off in a minute. Oh, hello, everybody. All of whom I know, all of whom I talked to earlier in the last week or so. Welcome to the 2025-2026 Devil's Gate series. This program started in 1995.
I was born in that year.
Yeah.
I'm 30.
I had decided that maybe I should give you a little background because there are a lot of you, I think, some of you who were born at or after the program started. Kathy was involved in the start because there was this picnic. And the picnic was Bill and Kathy Wilson, Bob and Connie Primus, Gary Long and Kathy Haight. And you guys took a picnic up Peru Creek. At which point in time you said, well, you know, we should know more about this area. Okay, so 31 years later, we know a little more than we did. And we started with very formal papers, like John Calhoun doing a presentation on theater. Then we got into slideshows once we had this, like Gary Long doing the program on privies.
which I made the changes flush.
Yes, so that when the pictures changed, it went flush. We have a lot of, well, we've had over 200 programs.
Wow.
I stopped counting on mine after 25. It's like, here, you do this. No, anyway, but it's really quite a wonderful amount of research and material that's out there at this point. Some of it is now out on the library's web pages. So if you go to the library's YouTube page, you'll find the most recent ones, like John's presentation on the spores, you know. And if you remember last year when we did cosmic ray research, which started with what is a cosmic ray. We're going to do Herbaria tonight, which I think is probably going to start with what is an herbarium. But it's been a wonderful combination of things that are out here, and we should all be very proud of what's happened. But our speaker tonight is Barbara Thiers. And let me tell you, this is great fun to have somebody from out of town. No. Barbara is former director of the New York Botanical Garden Herbarium, position she held for 20 of her 40-year career at that institution. She's currently an emeritus director and curator at the Botanic Gardens and a research associate at the Denver Botanic Gardens, which is how we came across her. For those of you who were here for Amy Schneider's presentation on Alice Eastwood, she came up with the interest in doing that after looking at Barbara's beautiful book called Herbarium, which is just cool stuff. And I'm going to let her talk more about what it is and what she's doing. But anyway, thank you all for coming. Oh, there's the can in the back for donations that then help us give some money to the community center. So thank you.
Thank you so much. It's a real pleasure to be here with you tonight, because this is a great center of, you know, Western botany right around here, as you know. I have to tell you of all of the botanical events that took place right around here. But we're going, and we will come back to that. I'm going to take you a little bit around the world first. We will come back to Colorado. So, for the past...
Better or worse?
No, that's great.
Can you see all right, Barbara?
I can see just fine, yeah. For the past almost 6 centuries, scientists have been documenting the plants and the fungi of the world through herbaria. This very simple method of preserving biological tissue by drying it, and in the case of plants, gluing them to a piece of paper, is a very simple technology, but it was key in transforming the study of plants from a minor subdiscipline of medicine into its own scientific endeavor. The herbarium made it possible for scientists to characterize those plants that grow in faraway places and also to understand diversity on a global scale. our wealth of specimens today that has been so carefully preserved over the centuries is a unique source of data that help us understand how the world's vegetation has changed over time and to predict how it may change in the future. So in my talk today, I want to tell you a few stories that I especially like from the history of herbaria, with a little extra focus on some stories about things that happened here in our own beautiful part of the country. Also talk about a bit about how herbaria contribute to our understanding of the environmental challenges we face today. And most of this is from my book that Christine mentioned. So evidence suggests that this man, Luca Ghini, created the first herbarium. He was from the region around Bologna in Italy, and he lived at a time when the Renaissance was just beginning to blossom in northern Italy, and forward-looking universities were encouraging independent thought, and new ideas and goods came from travelers who were passing from Europe to the Middle East and beyond and back. And after he graduated from the university, with a medical degree, he was hired on as a professor, and he offered what is generally considered to be the first botany course, and it was a course in identifying medicinal plants for the medical students. And it was a very popular course and eventually became part of the core curriculum. And instead, very characteristic of the Renaissance time, instead of focusing just on the classical texts like Dioscorides, which was centuries old but was still used at that time for how to identify plants, he wanted to make independent observations and have his students do so as well. So he went out and collected plants and preserved them by pressing them for his students to study. And then he glued them, the ones from one year he glued in the pages of a book, and then the next year he added some more. Voila, that's how that's how the first herbarium began, as a teaching tool. He later went on to create the first botanical garden in the city of Pisa, and again, that was also for the medical students so that they could see plants were living and so they could learn how to use them for medicine. Botanists came from all over the world to visit Ghini and to learn about this technique. And his herbarium itself does not exist any longer, but those of some of his students still do. These are from the late 1500s, when they've been very carefully preserved. The most beautiful of them is the one by Leonard Rawolf, who was the first European botanist to do collecting in Turkey and the Near East. And his volume, he pressed the plants exquisitely, and he around the edges of the pages, he used a kind of wallpaper that was used to cover beams in a wooden building, just to either decorate the sheets or maybe to just create a little bit of space between the sheets so the plants wouldn't get crushed. This is a very sumptuous volume which was owned by a series of different European royalty, and eventually came to be housed at the Natural History Museum in Leiden in the Netherlands, where it still is. And most of these others are also in very similar, like very old archives of Europe. So at the time that Ghini was inventing the herbarium, other world cultures were building upon centuries of plant knowledge that really surpassed Europe in terms of volume and continuity. Chinese scholars produced a new volume on plant knowledge at least every century, beginning in 300 BC and continuing, for centuries and centuries. And that led to a kind of an unbroken source of knowledge and consistency in the knowledge of plants and their method of naming them and illustrating them. And this was also happened in the Islamic world, where the plants of the Quran were very important. Plants of paradise in the Quran were very important for people to know, and they assumed they were the same plants that occurred on Earth. So again, there were many, many books over the years which refined over time the knowledge of the plants. And a very similar situation also in the Aztec culture, where over many centuries, information about plants were recorded, and they had supposedly their compendia and gardens that far surpassed what we knew in Europe. Most of that knowledge was erased when the colonial powers came in. What we do know about the Aztec knowledge of plants comes from a couple of monks who were Aztec, but converted to Catholicism and wrote everything down. It's called the Botanical manuscript. But anyway, none of these cultures, they had great plant knowledge, but none of them developed herbaria. And it's curious, but partly they were more temperate climates, unlike Europe, where the plants obviously change very drastically from winter to summer. In some cultures, the plants look the same more for all year long. That could be one reason. But also, they just may not have felt the need. I mean, the herbarium came about as a Renaissance innovation at a time when people were reinventing things. They were sort of saying, we're going to start anew and make fresh observations on the world and not count on what the Greeks did 10 centuries ago. And that's why we have the practice of dissection of corpses to understand human anatomy. It's where we got the telescope and the microscope, and also probably natural history collections, all in the idea of direct observations in order to advance knowledge. Beginning in the 16th century, European countries began to invest heavily in sea and land-based exploration, the goal of which was to establish colonial routes for sending goods back and forth, exploiting the resources of other outposts and bringing those goods home for sale. And many of the products they were keen to obtain were plant-derived, most notably spices, as I think we all remember from middle school and high school, but also horticultural plants like tulips, which were sold for astronomical prices in the 1600s. And although, so many of the expeditions, the botanical objectives were goods for commerce, there was also the idea that, well, we should be looking for other plants. We should be seeing what grows there. Who knows what new treasures may be living in these foreign lands that we could benefit from, but we just don't know about. Europe has something like 12,000 species of plants, whereas there's more than 400,000 species of plants known worldwide. So it's very, very common for these expeditions to bring back plants that no one had ever seen before. And that's, these began to be, when they would bring them back, they would dry them and press them and bring them back, and they were added to herbaria. So the people that were charged on an expedition with making plant collections, the way they would do, they'd go out from the ship, they'd see a plant that looked like a good one to collect, and that would usually mean that it had flowers or it had fruits. It was it had many of the features that you'd need to identify the plant. And then they would collect those and then press them. You do this step of arranging where you flatten the plant out. We use newspaper today. They used pages of old books and things back then. You flatten them out, try to make it look so that you can see the parts of the flower that you need to. And then as quickly as you can, you sandwich it between absorbent paper, and then you press it between two pieces of wood with a strap or with screws or something like that. That methodology has hardly changed. We still do it exactly the same way. And then, depending on where you are, you would either air dry it or you would apply some heat. This is an expedition in the tropics, and there are propane stoves under those that are bringing the air out. But anyway, once you've done that, as long as you keep the plants dry and keep them from breaking, they can last practically forever, certainly long enough to survive a sea voyage. And then once the plants are brought back, the step is to mount them, we call it, where you take some stiff paper and glue, usually. You just put glue on the plant in one way or another. Sometimes they're sewn on and arrange them. And then another, just as important as the plant really, is a label that tells what it is, where it was collected, when, and by whom. And so those are all the parts. So the style has changed. Now, of course, we print these. The old days, there was a lot of old handwriting that's very awkward to read. But there's very little change in how we do it from the way they did it 300 years ago. The first explorer that was specifically charged with the collection of plants on an expedition for scientific purposes was a man named William Dampier, who started his life at sea shortly after he completed his elementary education. He was an Englishman, and as a young man, he worked on ships that harvested logwood, which is a tree that grows along the shore of the shores of the Gulf of Mexico, the Caribbean Sea there, and the bark is used to, shredded and used to make a dye that was very, very popular for the color purple back home in England. And later he went around the world as a privateer. And a privateer was sort of a government-sponsored pirate. They had license from their government during a war and a conflict with another country to attack a ship and take its goods, and then all of the whatever goods were on the ship would be split between the privateering crew and the government. So so he did that, but he was unlike many of the people who did that in that he kept a very careful journal of everything, not just what he did and what he saw and what he pillaged, but also landforms, animals, sea currents, weather. It was a very precise and lucid account of every place that he went, and he would write these on long sheets of parchment and then store them inside a bamboo stem to keep them safe. And when he got back, he wrote a book about his travels, and it was called New Voyage Round the World. That was the title page. It was immensely popular. It went through several printings. It was translated into Dutch, French, and German, and it caused quite a sensation. And he went from being a lowly privateer to being somebody who was meeting all of the kind of the big thinkers in England at the time. And that got him the chance to get his own ship, a legitimate ship, to do exploring. The Royal Navy wanted him to explore what they called Terra Australis, which was Australia, but they thought Australia connected up to Antarctica. They thought it was all one piece of land, and they wanted him to go all the way to Antarctica on the ship. And it was great, except that the ship that they assigned to him was this one called the Roebuck. It was an old ship, and it really was at the end of its useful life. In fact, it was slated to become what they called a fire ship, which meant in a sea battle, you'd fill an old ship that you didn't care about anymore, full of explosives, and then send it into the enemy, fleet, and then it would blow up. So it tells you how much they cared about it. So he was a little disappointed about that. But anyway, he took off and he headed south from England. And unlike most expeditions which went that way, which went around the base of South America, he decided to go around the Cape of Good Hope and go this way and take advantage of the roaring 40 winds that would be pushing him basically to Australia. And it was a smart choice because he made it to the eastern coast of Australia, around here-ish. Well, actually here is where he first landed. In about 4 months, he did not have a single casualty, not a single case of scurvy along the way. And so, they made their first landfall on what they called a little island off the coast here. It's called Dirk Hartog Island. And they were running low on supplies, but everybody was healthy. And he collected about 20 different plants. This is what the vegetation of I have a hard time saying that, Dirk Hartog Island looked like. It's very low vegetation, but he made about 20 different collections, and most of them survived. He pressed them between pages of a book somehow. And in another landfall, he was the first person to collect this stunning plant, which is a very low-growing but beautiful plant. The flowers are about that big. It's called Sturt's Desert Pea. And so then they made their way up the coast, and this is his specimen of it from 1699. So again, it's been saved all this time. The idea was to make their way then around the coast atop of Australia and then get to the east coast. But long before he got that, he was only to Timor, which would have been just a little bit. You can't see. I don't know why I'm thinking I have another slide of it. I don't. But anyway, to Timor, which is just off the coast of Western Australia. And he found that the ship was so badly damaged by shipworms that he decided to go back. So he did not reach the destination he was sent to do. He turned around and headed for home. And he made it most of the way, but the ship did sink off Ascension Island, which is off the coast of Africa, northern Africa. And again, everybody survived. They were close enough to shore, they could all swim ashore, but he did lose a lot of his specimens, not all of them, but he did lose quite a few. And they waited there a few months, and then a Navy ship, Royal Navy ship, came by and picked them up and took them home. So the surviving plants were the ones that he made on Dirk Hartog Island and a few in New Guinea, and they were deposited in the Herbarium of Oxford University, which they were largely forgotten about for a long time. Eventually they were rediscovered. But 17 species were described based on his collections. He wrote another book about this trip, which was not as popular because it had less about swashbuckling kinds of things than the first book, but it was a key resource for Darwin, Humboldt. All the later explorers used his book, and many other sea captains did too, because of the very accurate descriptions of landforms, sea currents, and all sorts of things like that. And he didn't write any more books after that. He didn't do any more collecting, but he did make an enormous impact, including the fact that his books contain the first known usages of more than 1,000 words, according to the Oxford English Dictionary, including words that we use all the time, such as barbecue, avocado, chopsticks, and tortillas. All because of Dampier. So, a man with a huge impact, even though most people have never heard of him. Finding Terra Australis was also a goal of a very important French expedition called the Bougainville expedition, which was from 1766 to 1769, so almost a century after Dampier, and this was sponsored by the French government. A man named Philippe de Commerson was appointed as the naturalist for this expedition. He was a French landowner and physician whose passion was plants, which he'd been collecting and growing since he was a boy. It was probably in the course of his pursuing his botanical hobbies that he met a woman named Jean Barret, and they became lovers. Barret was an herb woman. That is, she made her living collecting plants to sell to physicians and apothecaries, and was quite knowledgeable about the local flora. And when Commerson was asked to join the Bougainville expedition, they told him he could bring an assistant, and so he chose Barret. Now, however, there was a royal ordinance forbidding women from sailing on French naval ships, and the Navy, the sailors were very, very superstitious, and they felt that a woman even touching foot on a ship was sort of dooming it to, some sort of terrible fate. So much less having one going along on the trip with them. However, they found a way around it. They had her dress as a sailor, and shortly before they took off, this supposed young man came on board, wanted work, and Commerson said, "Yeah, this could be my assistant." So she moved into his quarters, and they took off. It's a good thing for the expedition that they did, because Commerson actually was in very poor health. He had an infection in his leg, which he had when he got on board, and it never got better, and he couldn't do much field work. So it was really Jean Barret who did almost all the botanical collecting on this expedition. And they made stops all along the coast of South America, eastern South America. They went around the Cape Horn, and then headed to the Pacific. And they stopped for a while then in Tahiti. And it was a significant, the event in Tahiti was significant in the story, because this is where somehow Barret's identity was discovered. And the captain, Bougainville, was actually, he chose not to punish her or Commerson, but there's strong evidence that the sailors didn't take kindly to this, and she was subsequently raped and mostly kept to her quarters after that. And as the expedition was making its way back to France, they stopped in Mauritius, and that was a French colony, a logical place to stop and refuel. And a man there named Pierre Poivre, who was the founder of the Botanic Gardens, which still exist today actually in Mauritius, requested that Commerson come ashore and stay for some months to help him carry out botanical explorations. Well, fun fact that Poivre is probably the protagonist of the tongue twister, Peter Piper picked a peck of pickled peppers. Poivre translates to pepper in English. And peppercorns were a major export from Mauritius, and they were usually pickled in brine. So anyway, just for fun. But it was very convenient to Bougainville, whether or not it was really Poivre who wanted them to stay ashore, or whether it was Bougainville's idea. It was convenient for Bougainville because he was about to return to France to a magnificent homecoming after this expedition, which had really had very few problems along the way. But Beret was already, her pregnancy was already beginning to show in Mauritius, and there was no way that this was going to be kept a secret when they landed back in France. So Commerson and Beret got off in Mauritius, and they stayed. The rest of their collections went on home, but they stayed, and they spent 5 years doing field work. She gave birth. She gave the baby up for adoption to a French family living in Mauritius, and they had a very profitable time, made tons of collections over the next 5 years. But Commerson's health continued to decline, and 4 years later he died in Mauritius, and that left Beret without an income or a place to live. She had no government job of her own. But she's a very resourceful woman. She opened a tavern in the capital city, and the following year she married a former French army sergeant, and they subsequently returned to France, and she then became the first woman to circumnavigate the globe. Her homecoming was not officially noted, but she was granted a pension, and she also had a bequeath from Commerson. So she and her husband lived comfortably in his hometown in Saint-Olay, which is near Bordeaux, until her death at 67. And I went to France in October, Roy and I went to France in October of 2019 to see her grave. And I was delighted to see that it's well documented and maintained, and fairly often visited, judging by the number of offerings and things that were around. The specimens from the expedition, there were some 30,000 specimens, a remarkable number, and that would have been plants and probably animals, shells, and things too, which included 3,000 new species that had never been seen before, by far the largest number of natural history specimens ever made on an expedition. And most of them made by Beret. And they're stored now in the Museum of the National Herbarium in Paris, and have been digitized and can be viewed online. The most famous plant they discovered was the bougainvillea, which is named for the ship captain. And this is, of course, an extremely popular horticultural plant in tropical, subtropical climates. So this is one expedition. There were many. They all have stories. Maybe not quite John Beret caliber, but lots of trials and travails, but the plants kept coming back to Europe. And the fact that we could kind of make sense of these, that these could be organized physically and also conceptually, is really due to a man named Linnaeus, who I'm sure you've heard of. He's considered the originator of our method of naming plants and animals. And he was very much a larger-than-life character. He did not travel widely, but he still is absolutely key to any discussion of plant exploration. He had a breadth of vision, he had a compulsion to organize and to teach, and he also had a lot of self-confidence, as that quote would indicate. And this sort of gave him the power to construct this intellectual framework for classifying and naming plants. We no longer use his classification system, but of course we do still use his conventions for how plants and animals are named. And the infrastructure he put in place, it facilitated the incorporation of new knowledge, and it created enduring standards for preserving the physical specimens as well. And he wrote a book called Philosophia Botanica, which is considered the first textbook of botany. And he wrote in there about the importance of a herbarium, and he described how they should be arranged. He developed his own type of cabinet for herbarium specimens, and he had a diagram of how the plants should be arranged on the shelves. And at the time of his death, it was 1778 was when he died, his herbarium consisted of about 14,000 specimens, and it was purchased by an Englishman named James Smith, who brought it to London and sort of established the Linnaean Society and the Linnaean Society Library to house the specimens. It's still maintained. It's a beautiful building maintained in central London, and his specimens, which were originally kept in that cabinet I showed you, they're now in a vault in the basement, climate controlled, in these bundles. There's his library there. No expense spared in preserving them. And this is one of Linnaeus' specimens. So this is made in the, I don't know what year that one is, but mid-1700s. But it's beautifully pressed. And remarkably, it's been so carefully cared for that the blue of this delphinium, it's a larkspur, is almost as blue as one collected last year. So a remarkable degree of preservation. And again, these have all been digitized and they're all available online. So now moving to the United States, the first government organized expedition in the United States that resulted in herbarium specimens was Lewis and Clark's expedition. The goal, of course, had the objective of exploring the newly acquired Louisiana territory, and it opened the door to the westward expansion of the country, and of course the displacement of the native people who lived there. Both Meriwether Clark, Lewis and William Clark were trained to make botanical specimens by a Philadelphia botanist named Benjamin Smith Barton, sorry. And it was Thomas Jefferson who arranged for Barton to train them in this, because Jefferson was very keen that they make proper botanical specimens. And Barton, in return, was supposed to be responsible for identifying the specimens once they were brought back. However, Barton didn't do that. He either lost interest or was in ill health, and he never actually looked at the specimens. When they came back, they sat for a while and eventually were turned over to a German botanist who was visiting Philadelphia, a man named Frederick Persch. And he was in Philadelphia studying for a while, and he looked at the Lewis and Clark specimens, and he named some of them. He named a handful, which he named, and then he turned back over to the Academy of Sciences in Philadelphia. After the expedition, the main people involved, Lewis and Clark went on to do other military duties. Lewis got involved in managing Louisiana and so forth. He had a lot of financial problems, and he eventually committed suicide. But it was as if they'd never made these collections. They paid absolutely no attention, and this guy Persch kept saying, I've got these specimens, what do you want me to do with them? And nobody would tell him. So he went back got himself a job in England, and he went back there, and he just took the rest of them with him. Not without asking anybody, but honestly, there wasn't really anybody to ask. And he kept working on them in England, and under the support of a wealthy amateur botanist there, he was able to publish his work on North American plants, which was a 2-volume work in 1817. It was one of the most comprehensive floras of American plants that had been published to date, and it did include the Lewis and Clark specimens, many of them, including the new genus Lewisia and the genus Clarkia. When he was done with that work, he gave the specimens to his English patron, and went on to do other things. When his patron died, the plants were, his whole herbarium was sold by his family. They put it up for auction, and it just so happened that an American botanist named Edward Tuckerman just happened to be in the area at the time, went to the auction, saw what was being auctioned off, and realized, oh my goodness, this is the Lewis and Clark specimens. So he bought them, and then when he came back home, he donated them to the Academy. So they were almost lost to the United States for good. And for 40 years, these Tuckerman collections, which really represented less than a third of the specimens they collected, were the only ones that anybody knew about. They thought the rest had been lost until in the late 1800s, almost 1900, someone opened an old box in the attic of the Academy of Sciences, and there they were. There were the Lewis and Clark specimens in their original papers, like they had not been touched since they were sent back. And they weren't in very good shape. They were bug-eaten in some cases. The paper was in bad shape, but it highlights a theme that sort of runs throughout the history of herbaria, that the huge amount of effort goes into the collections, lots of fanfare about the expedition and the swashbuckling nature. But once the stuff is back, and it has to be dealt with, and the tedious part takes over, and there's not so much interest in it. And an awful lot of specimens sat around and weren't dealt with, and the Lewis and Clark is a classic case of kind of the mismanagement that took place. But the story has a happy ending because they have been resurrected. They have their own room at the Academy of Natural Sciences in Philadelphia. They're beautifully cared for. You see there's specimens, you can maybe see there's all kinds of little pieces of paper stuck all over them. That's all the people who have studied them. They're very well studied. Everyone wants to study them. So there's 222. There's still some that were lost forever, but it has a, we did right by them in the end. So after the Lewis and Clark, the U.S. sent many additional expeditions to explore the western U.S. And usually somebody, often someone who's also a physician, was charged with doing the plant collecting. And one of the first expeditions to visit Colorado, the botanist slash physician was Edwin James. And he served as he was on the Major Stephen Long expedition in the 1820s. And he was actually the first person to climb to the top of Pikes Peak. And for a short time it was called James Peak, but then they decided to honor Zebulon Pike, who I believe had just died, maybe died in the course of his, whatever he was doing militarily. And so they renamed it. But anyway, James was the first one there, and he collected, the first representatives of Colorado's alpine flora. And he also, not alpine exactly, but lower down, he collected the first plant of our state flower, Aquilegia cerulea. And this is that specimen that comes from his original collection and his label, which is sort of hard to read. But the specimen was preserved at the New York Botanical Garden, so I've had the pleasure of seeing it. And the reason it's at the New York Botanical Garden is because in the early days, there were sort of two botanical greats in the United States. There was John Torrey, who of New York City, and later his student Asa Gray of Harvard University. And so everything that, really most of the plants from those early expeditions came to one of these two men to identify. Torrey, who was a New York City native, who became the first really internationally recognized authority on American plants, an American who was an authority on American plants. He was never actually employed as a botanist. He was again a physician who decided he did not like practicing medicine, so he developed other skills. He taught chemistry and geology at West Point, Columbia University, and at Princeton. But the study of the North American flora was his passion, and he planned to write a flora of the entire United States. And so that's why he was keen to get all this expedition material that came from the Western expeditions. And his herbarium that he built from these specimens was about 80,000 specimens in size, very large. And when eventually he passed away, his collections went to Columbia University, and Columbia University's herbarium became the nucleus of the New York Botanical Garden herbarium. So that's why New York has so many Western plants. Now, Asa Gray was a student of Torrey's, an informal student. He was a very bright young man from upstate New York. He had his medical degree in hand before he turned 21. But again, like so many, so many botanists at the time, he had no interest in practicing medicine. And so he was interested in botany. He was hired by Harvard University as a young man and stayed there his entire collection. And he took over from Torrey as the person to receive the works, the specimens from all over the country. And the Harvard University Herbaria, which he founded, is 5 million specimens, is the largest university herbarium in the world. And his book, Gray's Manual of Botany, was the key botanical text. I think you can still get a copy. I think when I was in grad school, people were still using it. It had been updated by people since, but he was an incredibly influential person in North American botany. And of course, you know the names because Torrey and Gray's Peak here were named by another student of theirs, another physician turned explorer, C.C. Perry. Perry was explored all over the West and California, Mexico. He was one of the most prolific collectors of them all. Actually, I got ahead of myself, but anyway, this picture is to show that Gray, unlike Torrey, who never traveled, probably never went farther west than New Jersey, Gray did come to Colorado a couple times. And this picture, it's one of my favorite pictures of all times. It's from a trip that he made with Joseph Dalton Hooker, who was the director of the Royal Botanic Gardens Kew, a longtime friend of Torrey's. They were invited by Hayden, the Hayden expedition, which was an important multi-year expedition in Colorado in the West. They were invited to go on an expedition on the inaugural, that would take the first train trip. There was a new train line that was called the Denver and Rio Grande Railroad that went from Canyon City all the way to, well, it went from there to Fort Garland, and it went over Levada Pass. And Levada Pass at that time was a popular spot for people to go, I suppose, to get out of the heat and so forth. So they planned this expedition, and they all took the train from Canyon City. They got off at Levada Pass. The supplies for the camp, the tents and so forth, were brought either from Fort Garland or from Fort Lyons, right up there. And you see there's Gray there and Torrey, and I believe that's Hayden, and then a whole host of other folks. There's a guy in I swear, right out of Central Casting with a jacket with long, whatever, deerskin jacket and his gun. And they were interested in it because Torrey just wanted to see these plants in the flesh, and Hooker was a specialist in his early days in the Himalayan flora, and he was interested in the differences and similarities between the high altitude plants of Colorado, or the Americas, and the Himalayas. And I don't know if you've been to Levada Pass, but there are still some of the historic buildings standing, including the train depot where they would have debarked. Anyway, this is where Charles Perry, I have a picture of him, and I accidentally put an S on the end of his name for some reason. But anyway, he named a lot of these mountains for his botanical heroes, I suppose. And again, C.C. Perry's name appears on so many specimens as the collector. Hardly anybody has done more collecting in the West than him. But a short time later, Colorado produced its own botanical superstar. Alice Eastwood was born in Toronto, but moved to Denver as a young teen, and her love of nature, especially plants, grew during long hikes in the Rockies. And she was a talented student. She was the valedictorian of her high school class, but her family's financial situation was precarious, and so high school was the end of the road for her educationally. She went to work as a school teacher. But she still, even so, she continued to expand her botanical knowledge. She spent as much time as she could collecting in the mountains. And she developed her reputation for her knowledge of the flora and her knowledge of the trails and so forth. So when Alfred Russel Wallace, a very famous English biologist, visited Denver in 1887, he was on a speaking tour throughout the United States. It was the then 28-year-old Alice Eastwood who took him on a 3-day hike to the summit of Gray's Peak. And Amy Schneider from the Botanic Garden has done extensive research on this and knows exactly where they went. It's a fascinating fascinating story. They were lifelong friends after that. Russell was known for his work on evolution. He was really thinking along very similar lines as Darwin, but he was working in Southeast Asia. And anyway, Alice Eastwood, as I said, she eventually, for the real estate that her family owned, I believe is sort of in the Herbaria area of Denver, so where the college campuses are. And she sold that, made some money, and decided she wasn't going to work anymore and just pursue botany. So then jumping to California, although Torrey and Gray were the dominant forces in American plant diversity in the early 19th century, as people moved westward, botanists moved westward, institutions were established. And the most important of these in the early days was the California Academy of Sciences in San Francisco, which was founded in 1853 with the mission of documenting the plants and animals and geology of the state. The Academy had an interesting clause in its charter indicating that the institution, quote, highly approved the aids of females in every department and earnestly invited their cooperation. And it thereby became one of the first institutions in the country and possibly in the world to recognize and encourage women scientists, a decision that really paid off for the institution in spades due to the efforts of two highly accomplished and energetic women that it employed. Kate Brandige, who was trained as a physician at UC Berkeley, was hired as the curator of the Academy's herbarium. Again, another physician who really wanted to be a botanist. And she and her husband, Townsend, collected extensively throughout California and also Baja California. And she loved collecting so much that even though she was the curator of the herbarium, she decided she no longer wanted to do that. And in her place to do the curation work, she hired Alice Eastwood. So Alice left Colorado for San Francisco, where she spent the rest of her life. And it was under Alice's supervision that the Academy Herbarium became the most complete record of Western plant life and was frequently consulted by botanists all around the world. The Brandigues, although enthusiastic, were apparently somewhat negligent curators. Again, like the collecting, didn't care so much about what came after. So Alice spent a lot of time organizing and refining the collection so that it was easy to use and well preserved. Her energy and enthusiasm for the study of plants and her dedication to the herbarium gained worldwide recognition through her heroic efforts to save the herbarium during the earthquake and fire in San Francisco in April of 1906. She felt the tremors that morning when she was home, but it wasn't very strong in her neighborhood, so she went to work as usual. But by the time she got the Academy, she saw that the building was damaged, and worse, the surrounding buildings were all starting to burn. But undaunted, she wanted to save what she could of the herbarium, so she went into the building. The staircase was destroyed, but she climbed up the banisters somehow to the 6th floor. And from there, she gathered what were considered the most important specimens and her Zeiss lens, her magnifying lens, and she bundled them up, lowered them through the window, and then began to climb back down the way she came up. By the time she got to the ground floor, the fire was really closing in, and the firemen were desperately trying to get everyone out of the area. She found a horse and buggy to carry the specimens to safety, and got out herself. Later, her own home on Russian Hill was destroyed as well. She in a letter to Science magazine that was published a few months later, she wrote, "It's a great loss to the scientific world and an irreplaceable loss to California. My own destroyed work I do not lament, for it was a joy to me while I did it, and I can have the same joy in starting again." What a spirit. And when the Academy reopened its doors in 1916 at its new quarters in Golden Gate Park, Eastwood went right to work rebuilding the herbarium. She could indulge her interests also in education. She ran botany courses for the public and for the gardeners. And for the remainder of her long life, she collected plants to rebuild the herbarium. She built it up again to 300,000 specimens by the time of her 80th birthday. And she was offered many, along the way, many honorary degrees, but she refused them all because she just could not accept the premise that only somebody with some special fancy letters after their name could make a contribution to science. And she had little patience for academic conventions or for what was expected of female comportment and appearance. She wore she fashioned some sort of a split skirt so that she could hop on a horse and so forth. And when women would say to her, "Oh, I wish I could dress the way you do," she would say, "Scorn convention and you always may." She died at the age of 95, and they later named in the California Academy, they named the Allen Eastwood Hall Botanical in her name. So it's through the work of explorers and scientists, such as the ones that I've talked about, that today there are about 4,000 herbaria around the world that hold in about 194 countries. They hold together about 403 million specimens. They still serve their original function, which is to document the occurrence of plants and fungi and to provide a reference for identifying ones of unknowns. However, more recent technical advances have now allowed us to study life at both the molecular levels and on global scales, and these technologies can be applied to herbarium specimens as well to help us address some of the more critical environmental problems that we face today. So understanding global change on Earth's organisms and ecosystems is obviously a critical focus of biological research today. But documenting such change is tricky because it requires measurement and observations of before and after. And honestly, we don't know exactly what was before, except in the case of specimens, we have the actual tangible material of things that lived long ago, and we can compare them to what lives today and see what we can make of the differences that we find. They provide one of the few tangible sources of data about how plants and fungi lived before industrialization and through each successive period of technological advance at that time. Because plants are rooted in the ground, they're especially exposed to the conditions in which they live. Case in point, heavy metals, we know, are found in soil as a result of industrialization, pollution, agricultural runoff, and so forth. And these can have adverse effects on animal life, not so much on plants. Plants tend to concentrate them, but they don't tend to kill them necessarily outright. And so we can look at specimens from periods of the past and we can assay them for their heavy metal content. And indeed we can see, it confirms what we know, is that over time, since we've put better practices into place, we find fewer and fewer heavy metals than we used to. But it's interesting, for example, a study done out of Brown University showed very high concentrations around all of the industrial plants that were in the East Coast, and it was obvious that there was an industry there. But 13 miles offshore is Block Island, which never had any industrialization. But plants from that period, from Block Island, show equally high concentrations of heavy metals, even though there was no industry there and there was no outward signs. So plants can tell us something that we can't see with our eye about what was going on in the soil. In the 1900s, scientists discovered that gene sequences could be recovered from the tissues of herbarium specimens. And over time, that process has been refined so that you can use smaller amounts and you can get DNA from older specimens. The oldest specimen that I know about from which DNA has been extracted is a specimen from 1828. So you can do it. It's not easy, but you can get DNA out of very, very old specimens. And until recently, most of the reason we did that was really we're looking at trying to reconstruct a family tree of herbaria. By comparing gene sequencing, seeing which ones were the most alike, that would mean those plants are most related, and we can construct a genealogy to understand plant diversity. But now, there's more powerful tools. You can get really a lot more information. You can study more details about the gene sequences, and you can understand a plant's genome, its entire genetic makeup. When you can do that, you can begin to understand what genes are controlling features like nitrogen fixation, like what genes allow a plant to fix nitrogen, which ones allow it to be drought tolerant, what genes make a plant invasive and not another plant invasive. And so this is very powerful, and for whatever reason, herbarium specimens are extremely well suited to extracting genomic DNA, because the first thing you have to do when you study genomic level data is you have to break the DNA up into many different little pieces. And when the plant dries, it's already broken up into pieces, so you kind of save a step. So there's a lot of work going on right now looking across vast numbers of plants to try to understand things like, what genes have led to features that will allow us to grow plants in areas where we can't grow them now, develop strains that can grow with less water, that can tolerate more salinity, and so forth. And herbarium specimens have a lot to do with that research. Specimen digitization, I've mentioned this many times already, but this has been the process where you take a picture of the plant and you take the information from that label I showed you and you put that into a structured database, and then you can search for plants of a whole host of different characteristics, and you can see the specimens. You can plot where they exist on a map using geo-coordinates. And during the last decade, 15 years or so, there's been a massive effort worldwide and in the United States to digitize as many specimens as possible. And now I think there's about 80 million worldwide digitized specimens that you can just, for free, anybody can just call up and look at. And the National Science Foundation funded a lot of this work in the US. And this has opened up many new avenues for research. You can plot things on a map and see how they relate to other factors. I throw this up here. In 2019, there were terrible fires in Amazonia. And so we made a map of the specimen, all the Amazonian collections that had been made, the dots. The green is the forested areas, and the red is where the fires were burning. And so we could see, what species were most impacted, and because many species have very narrow distributions, to develop ideas about what species might have gone extinct as a result of those fires. When making herbarium specimens of plants, collectors are, as I think I mentioned, they're very keen to get the features that will help you identify them, the flowers, the fruits, and the buds. So most herbarium specimens have these features. And these structures also indicate what we call the plant's phenology. So what time of year it forms a bud, what time it flowers, and when it makes a fruit. And we can tell that because the specimens always have a date on them. So specimen collected in 1960 in April, we know that the species was flowering at that time. And now with all the millions of digitized specimens that we can look at these collectively and see if there are trends over time. Can we determine that in some cases plants are now flowering earlier than they used to because of a warming climate? And it's, there are cases, especially here in this high altitude, where it's pretty clear that plants are flowering earlier. There have been any numbers of studies just here in Colorado. This is a big focus of the work down at Rumble. A lot of people are doing this kind of phenological study. And we care about this because this means that there could be a mismatch with the pollinators. So an insect pollinates a plant, the insect comes out, it starts its life cycle, reaches maturity at a certain point. Insects and plants have co-evolved so that the insect becomes of an age to want to collect pollen just at the time that the plant's producing pollen. And if one of these gets out of sync, then either the flower doesn't get pollinated or the bee doesn't have anything to eat. And so again, there's beginning to be some pretty definitive documentation of these mismatches happening. And again, herbarium specimens can give us a clue as to where the problems might be. So because of all the digitization and the expansion of research, the past decade or so has really been a golden age for herbaria. But still, we face significant challenges at the moment, most stemming from insecurity of funding for these collections. Herbarium budgets rarely grow in good times, but they almost always fall when economic times are bad. And the pandemic was a really hard time for everybody, of course, and it was a bad time for collections and museums in general because of reduced revenue, and funding has not really returned to pre-pandemic levels. And in our current climate, there's reason to be concerned about the future of some herbarian and natural history collections. So we're worried, these collections have to be maintained in perpetuity to be of value. Oh, I'm sorry, even in the, accidentally pressed the wrong thing. Even in the best of times, we're always worried because to be of value, these specimens, as I said, have to be kept forever. And it's a huge commitment and a leap of faith for an institution to agree to care in perpetuity for a collection that is ever growing. Collections need care. They need to be kept from fungal and insect damage, and they need to be kept at the right temperature, and they can't be flooded and so forth, and they have to be available for people to study. So in order to justify keeping these collections, the onus is on the curators and the people who take care of the plants to keep coming up with ways to make them relevant to the work that we do. We have to continue to play a role in botanical education and provide service to the scientific community and continue to come up with new ways to extract scientifically meaningful information from this 500-plus-year-old record of plant life so that can help us, sustain the ecosystems that keep us alive here on Earth. And that's that. Thank you very much. Happy to answer any questions. Oh,
Sorry. Okay, so regarding, for instance, any herbarium throughout history is plagued with bugs and fungi infections or invasions, and then also DNA preservation. So throughout the history of herbarium, have there been practices and tactics and tools that you are now using when framing future herbarium prints so they can be preserved better for people that study them in the future and so forth?
Well, yes. I mean, as I mentioned, the basic techniques are pretty much the same.
Right.
We use archival paper so
Yeah.
that the paper does not acidify the collection, doesn't disintegrate.
Yeah.
It's important.
Has there been any tools that are made specifically for herbarium, like any glue or adhesives?
Yeah, I mean, not really.
Okay.
We use a white Elmer's type water-based glue,
Sure.
which
that's
is also
what I
archival,
use, yeah.
and 100% cotton rag paper. We use archival, whatever the archivists recommend. That's often what
Sure.
we use.
Sure.
The cabinets. If I go back one slide, you see the steel cabinets. These are specially designed herbarium cabinets.
Is the room refrigerated at all, or
It's
temperature?
kept at about 50% humidity. Ideally, it would be maybe 60 to 65 degrees temperature. That's a little cool to work in. Most places bump it up to 70. It would really be better to be a little cooler. But these cabinets are steel. They have a rubber gasket all the way around. They close very securely. That's what everybody uses. There's a steel cabinet that prevents fire. Insects really can't get in there, although I'll tell you, insects do come in. The cabinet's open, and we have an extensive monitoring system for pests. All sorts of different pheromone traps and things like that. And those are routinely checked, and they do spot checks.
Do you smash them and glue them on paper?
I've seen some glued to paper. I've seen some bugs
Like
get
the
trapped.
grasshopper?
Yeah, no, I've seen it.
Grasshopper?
But what you do in that case is you freeze them.
How about fungus nowadays?
Fungus infection. So again, that's where humidity comes in.
Sure.
I've certainly seen herbarium specimens that have been damaged by fungi. That's much more of a problem in the tropics.
Yeah.
But it's why we try to keep them at a low humidity so that the fungi can't grow. Fungi are a little harder to control than insects. Insects will die if you freeze them. We're not entirely sure that the fungi do. So people used to treat them. They treated specimens with all sorts of horrible mixtures of mercury and so forth. But of course, that's terrible for the people who work there, so that's not allowed anymore. But they did used to sort of dip them in all kinds of awful concoctions to try to keep the bugs So, but you know, this is an active area, and every year when I go to conferences, there's always a session, very well attended, on the latest, you know, thinking about preventing insect damage, new archival products and techniques, new ways of protecting things. So
Fibers of paper, probably.
exactly. And you know, we always have at our conferences vendors of things people who are trying to, you know, promote new products for better preservation. Plants are easier than many other natural history organisms, but still, it's a constant battle.
Are you still coming up with lots of new, never seen before specimens? I think of the 19th century and just what
Yeah.
must have been showing up.
Well,
But
it's nothing like that
Yeah.
anymore. But there are still, every year, there are still new species described, usually from Southeast Asia or South America. But even in North America, occasionally a new plant is described. Now, my personal area of expertise is on bryophytes, mosses, and related plants. And there, again, that's more common because they're very small and often overlooked. And fungi, I don't know, Roy, how many fungi are described new each year? Hundreds?
Yeah.
You know, we're not even close with the fungi to knowing how many, you know, there'd be, I mean, here in Colorado, on any given year, you could easily find something that's never been described before. So yes, but not as much as we used to.
Do you find that that you see plants that have morphed from climate?
Yeah, that's a very interesting
Influences.
question. Yeah, and probably, so here's one case I can tell you about that where morphing is taking place. You're all familiar with cheatgrass, the horrible
Oh yeah.
scourge of the West. Well, it's originated in Europe, and it's not invasive over there. It's very well behaved. And so the question was, what happened to it? Why, when it got here, did it become the scourge that it was? And we happen to have in New York some of the very first specimens we think that invaded, because people were looking all the time in the 19th century. People were very avid plant collectors. Someone saw something they'd never seen before, they collected it, and they said, what the heck is this, to all their friends, and then they made specimens of it. Well, so a fellow at Washington State then did genomic level research on some of these very early collections, and he could trace collections from different parts of the country where it had entered, probably through shipping ports, back to the original locations in Europe. And it turns out that they weren't, in Europe they were from very different genetically distinct populations, not close together at all. But once they got here, all of these different slight genetic variants began to intermingle. And that's what the genetic mixing of these different strains, which somehow created this super plant, which is, you know, caused what, billions, I think they estimate billions of dollars in you know, damage to agricultural lands and grazing and so forth like that. So yeah, it morphed. You wouldn't see it. I don't think the plants look any different, but on a genetic level, they're very different.
Have plants that you know have morphed as a response to climate change?
Not that I know. I mean, that would be hard to say. I don't think we would have enough evidence yet to say that. You'd really need many years of data, and you'd have to really understand, if you're seeing changes in the plant, what caused that. You know, I imagine people are looking for that, but I don't know of any case where someone says, "Oh, yeah, I used to have yellow flowers, but now with climate change, the flowers are pink." You know, I don't think I've heard that yet. But you know, it doesn't mean it's not happening. It just hasn't been documented.
Yeah, I was just curious, like if I were a scientist and I wanted to do a DNA study, like, would I just walk into their herbarium and like just pick what I wanted and take a flick of it and like
Well, you
disappear with
probably
it,
have to
or
ask
would I-
permission.
There's a whole process for
So
that.
it's very common. People would say, "I would like to sample DNA from the specimen,"
Yeah.
and you go and you look at the specimen, and if it's ample, you know, if it's very old ones, often are just a few little stems, and then you probably say no. But if it's an ample specimen, you'd say, yeah, you can have, because it really requires just part of a leaf or something. So then you would give it to them, and they would take it back and they'd grind it up and get DNA out of it. Ideally, then what they do is they submit the sequences to GenBank, so everybody, and they document where it came from, and then they return the sample, whatever's left over, back to you, and you can either store the DNA, or you can say, no, you keep the DNA, but just give me the information. So yeah, where did you want?
Just to elaborate more on the question she asked. In the porcini family of mushrooms,
Yeah.
there was a botanist or mycologist in New York State describing an iconic species. Well, it turned out a lot of things looked like. So a whole bunch of diversity was called this one species. So we decided, what I was asked to do, ask the state herbarium in Albany if you can take the type specimen,
The original one,
the original one,
that the
the
person.
name was
Yeah.
made from, and extract DNA. Can we do that? Well, yes, there's, we'll sign this, you can sign that, and we were fortunate, we were able to do it. And so, and as Barbara just described, the sequences went to GenBank and say, if you have this species, it has to match this genetic identity. And now that it's easier and cheaper to do, extract DNA sequence and so on, we were doing that, and they go, oh, no, don't have that one. They don't have that thing that described from Southern New York.
So then the knowledge is not lost in this gene bank?
No,
No,
the only problem is people aren't always as careful as they should be about saying where the, where they get the DNA from.
Yeah.
There are lots of mistakes. But theoretically, there should be a trail back to the source. It's really not science if there's no trace back to the source. That's kind of a fundamental principle. And there are people who should know better who make these mistakes. But yeah, theoretically, when anybody who sequences any collection that I was involved with, if you sequence from a specimen, you can use it for your research, but then that is to become public knowledge and usable by anybody else who wants to use it. You don't own that, and you can't keep that information secret. You can't develop a patent or something based on it. It's for the public good.
and I had to return the DNA
Yeah, in a little vial.
in the vial back to Albany.
Yeah.
I'm glad to know how they do it. I have visions of the swabs that you do.
Yeah, they use a blender. That's what they do. You got a question?
I do, but if yours
Oh,
is
just
turned
like
out-
real quickly, like kind of like in the realm of her question, do the prints ever retire, like, and then they have to, at some certain point, like from the 17th century, they retire and then they have to make a whole new one, for instance, like after it's been picked at a lot?
We never throw anything away.
Sure.
There are sheets that have been so badly sort of abused that
Yeah.
there's just a fragment left. But you would want to know, you'd want to maintain that, at least
Okay.
keep it, even if it's really not useful, so that everybody else knows.
Okay.
So they're never like replaced or retired, they're just kept, which is why this gets to be a very expensive business. After a while, 'cause you can never throw anything
And that's why
away.
it's all of those, Wow.
Yeah.
So my two questions are maybe way peripheral
It's
to
okay.
the current discussion, but, and they relate somewhat to your question about financing and
Mhm.
things. So the first question I have is whether and how much this research was related to a desire to determine medication used for human conditions. So that's kind of one area. And then the other is more advanced climate change. My area of peripheral expertise is in forests, which, are they plants?
Yes.
And if they're plants, whether and how much there might be research on their carbon emissions
yeah.
and the balance
Yeah.
they may play in climate change. So
Yeah, okay. So the first one, the whole connection to medicine was the original reason to make a herbarium in the first place.
Okay.
And honestly, still, people use, there's a whole area of sort of ethnobotany, where people are still looking for new plants that that may have compounds that are useful in treating cancer and so forth. The golden age of that was 20 or 30 years ago when people were exploring all over the world. They ran afoul of many other countries who were saying, well, wait a minute, you're going to take this and make money? This is our plant, you know. But still, a lot of that is done, and Taxol is a classic case of where it was just, you know, Taxol, the drug that's useful in breast cancer, was taken from the taxus, from the yew tree, and that was discovered in the course of just like collecting plants and then just assaying them for chemicals. We used to do that. We used to, the New Botanical Garden, we would collect sackfuls of things from all over the world, and then we would grind them up and they would look for compounds. And never told us if they got any hits, and we weren't, just like the people in the countries who the plants came from weren't entitled to any of the profits, neither were we. That was all proprietary. So it's very highly controlled now, but it does still go on, and we're still learning because most of the drugs that we use, you know, many of them have a plant origin.
Penicillin was an accident.
Penicillin was an accident, a lab accident. Yes, absolutely.
I got that from the Big Bang Theory.
Yeah. And in terms of carbon in forests, where herbarium would come into that would be a little peripheral. So they would tell you the names of the plants that occurred in the forest, and they could tell you that the plants that used to grow there, maybe there's species that used to grow there that don't grow there anymore. That's a clue. It's nothing definitive, but it's a clue that something may have changed. But in terms of carbon output from a specimen itself, I don't think. Now maybe somehow by grinding up the tissue of a plant, you could get some kind of you could get some kind of signals about their productivity, possibly. I don't think it'd be the most efficient way to go about it. You know, I think you'd be much better off taking your machines out into the forest. But the problem would be with old forests that don't exist anymore. You know, that there you might want to take a more forensic approach. And maybe you would find something. Maybe there are people doing this. I'm trying to think of research that I know of that's being done like that. I don't really know that. Yeah.
So I would say that there is a movement in the United States to take better advantage of our forests in addressing the climate
Yeah.
balance.
Right.
So if there is a potential role to add to whatever research is done,
Yeah.
that might be something.
Well, that's what we have to keep looking for.
Yeah.
Ways that the knowledge of the history of plants, you know, and how they lived on Earth, how that can inform research that's going on
Right.
now. It just takes somebody clever to figure out a way, you know, to make the connection.
Yeah. And
That's really key.
That's what I'm all about, connecting dots.
Yeah.
So anyway.
Yeah,
thank
we
you.
often in science, even today with social media and internet and instant connectivity, there's still a lot of siloing going on.
Yeah.
People
Yeah,
don't look, they look straight ahead, they don't look to the left, and they don't look to the right. So yeah, it's a challenge to get people to do that, but powerful things happen when they do.
Thank you.
Sure.
Lastly, okay, real quickly, does herbarium have to do with like the collections of like seaweed too? Okay, so
absolutely.
is that like aquatic, or is there any barriers to that? Like,
No, they, so seaweeds, they're very challenging to make as specimens because if you basically what you do is you go to the rocky shore, you collect some seaweed, you also collect some seawater. They have to have the same seawater that they're growing in.
Sure.
You bring that back, you put it in a flat pan, and you lay the alga out, and you try to flatten it out, which is challenging in itself because they're all branched and everything, but you flatten it out. Then you take the sheet of paper, and you put it under that, and you lift it up. And then you dry it as you have to change the blotters, you have to dry it as fast as you can because it will begin to rot so quickly. And you can, well, as has happened in bitter personal experience, it's a beautiful red alga, it just looks like a coral, it's so beautiful. You lift it up and you think, oh, this is gonna be magnificent. If you're not careful about how you dry it. It's just black and it's smelly and really awful. So there is a real art to making a beautiful seaweed specimen. But there are some absolutely stunning ones. But it's a labor of love.
Because your focus is algae, so you're familiar.
Bryophytes. I work on
Your
mosses
bryophytes.
and-
Would you come back sometime and talk about
Sure,
bryophytes?
I'd love to.
Oh, okay.
I'd be more than happy to do that.
Well, I think it's something that we all just walk by all the
Yeah,
time, and
yeah.
you're seeing it at a depth
Yeah.
that most of us
This is a
aren't.
great area for bryophytes.
Yeah, we have
a lot of Do a talk and have a walk or something.
Yes,
Yeah,
absolutely.
Sure, we'd love that.
Barbara, thank you.
You're
very
welcome.
much.